Well, good morning, AP Bio students. Mrs. Shelton here, and I'm going to give you a quick overview of Chapter 14, which is gene expression. Gene expression, talking about how we get from DNA to the polypeptide. To give you guys an idea, this is probably one of the top 5 most important chapters that we're going to cover this year. Absolutely imperative that you understand transcription translation, differences between eukaryote and prokaryote transcription translation. Pay attention to the you must knows and really follow along, but it's going to be really helpful for you guys to make sure that you're reviewing this information in the test prep review series. So it's topic 5, and again, even in there, they're telling you that this is one of the most important chapters for you to pay attention to. And then from the old textbook, study guide chapter 17 is going to be incredibly useful for you too. All those interactive questions are really going to help you understand those differences in transcription and translation and how to actually decode DNA. Now, make sure that we are remembering that last chapter we focused on DNA replication. Now, in replication, we're copying DNA to DNA. This has to happen before mitosis and meiosis. In this chapter, we're talking about how DNA is used in the somatic cells in order to read the DNA to make RNA or also proteins. Because also remember, DNA serves as a template for all the RNAs that are used in a cell, and the DNA serves ultimately as a template for all proteins that are made inside the cell as well. So this is what we need to focus on. It's really not difficult, but it's just absolutely imperative that we understand these differences. So let me go ahead and switch over here to the presentation. We'll go ahead and talk about this. You're going to want to have your notes out in order to make sure that you are identifying these terms and making sure that you're comfortable with these terms. Let's go ahead and switch this here. I'll get this to full screen if it'll let me. See if we can. Oh, hold on. Patience. Present review so we can get this going. There we go. Okay, so what you are seeing here is, come back to me. There you go. All right, so what you guys are seeing here is just an illustrative example of how a change in one gene can change the phenotype of an organism. So this is just showing you an albino mule. And so in this, it's just one simple mutation, one change in the DNA for the gene that codes for pigment was altered, and so therefore this results then in a change in phenotype. So this is the important connection we have to make, is how those changes in DNA can result in changes in phenotype, is what we're going to need to make sure that we're addressing. Now, in this particular chapter, what we're really trying to make sure that you understand is the one gene, one polypeptide hypothesis. So every gene, when we talk about a gene, those segments of DNA, they code for one protein. Now, recognize too that for eukaryotes, for us, we have about 20,000 different genes, but we don't make only 20,000 different proteins. We make over 100,000 proteins in our body. So this is going to be an important aspect of being able to explain how we can have fewer genes but more proteins. And this becomes a difference in eukaryotes versus prokaryotes. In a prokaryote, the number of genes they have is typically the number of proteins that they can produce, the max number. Now, a lot of what our genes code for is for enzymes. Remember, enzymes being proteins, and they control our biochemical pathways. Now, what I do need you guys to make sure that you are seeing here, this is what is referred to as the central dogma of information flow in life. Information for making proteins is encoded in DNA. But in order to get to those proteins, that information has to be transferred into RNA molecules first, and then RNA molecules then actually will get translated into those proteins. Now, it's also these steps. I mean, we know that DNA is a more stable molecule. DNA is a double helix. RNA is a more unstable molecule. It's only a single strand. This actually is argument for why it is believed that life on this planet actually began as an RNA world and not as a DNA world, where RNA was the template for making proteins and very fast reactions, very fast lifespans, etc. But now this evolution to DNA made it a more stable, more consistent way to be able to pass information on. But we'll talk about that in our evolution unit as far as the understanding or belief that life began in the RNA world. It kind of makes sense. If RNA has to be the molecule that directly is translated into a protein, well, that biochemical process probably happened first. Now, do you remember the basic difference between DNA versus RNA? DNA being a double helix. Remember that the sugar here, deoxyribose, is a more stable sugar. Remember that the bases, G and C, A and T, are complementary bases. So do remember that thymine is used in DNA. Now, RNA being single-stranded, this actually gives it a tremendous versatility. And so in this chapter, we're going to be talking about 3 different types of RNA. So our messenger RNA, our ribosomal RNA, and our tRNA. But we'll also kind of introduce to you some other RNAs, like our snRNAs that are used for RNA processing, etc. So this single strand actually allows it to fold in different ways and be functional in different mechanisms. But RNA uses the sugar ribose, and with this extra oxygen, it's a more reactive molecule, so it doesn't last. Now remember too that uracil is a substitution for thymine. So if you're ever reading any RNA sequences, it's going to be really easy to tell because there's going to be U's instead of T's. So that makes the complementary base pairing. In DNA is GC, G and C, A to T. In RNA, it's going to be G and C, A to U is what's going to be happening there. Now, this is a quick overview to show you the basic differences between protein synthesis. Now, protein synthesis involves transcription and translation. Now, transcription is always the copying of DNA, just the DNA gene, into messenger RNA. So just a single strand of messenger RNA. And then that messenger RNA has to get translated by a ribosome into a polypeptide. And we'll talk about that. So in a eukaryote, we actually have RNA processing. So, and where translation happens is out in the cytoplasm, but transcription actually has to happen inside the nucleus. So DNA is going to get transcribed into actually pre-mRNA. We can see that there's a different process after transcription. There's RNA processing that has to take place in a eukaryote, and we'll discuss this more. And then my messenger RNA leaves the nucleus in order to now bind to a ribosome where translation can take place and the polypeptide can be made. Now, again, eukaryotes don't have a nucleus, therefore there's no separation of the location of where transcription and translation happens. It all kind of happens at one part, but transcription and RNA processing for a eukaryote has to happen in the nucleus, and then translation has to happen out in the cytoplasm. So there's our differences to make sure we have that. Now, when we do talk about a gene has to get copied, and so here's actually just kind of a blown up picture trying to show you that if this is my DNA, we do need to recognize that DNA being double stranded, only one side of it is actually the template, the gene that needs to get copied or transcribed. So we need to make sure that we're really comfortable with that word transcription. So when we say transcription occurs, we know that it's the DNA template strand that is now getting converted into a messenger RNA, so mRNA transcript. Now, the messenger RNA, and we'll talk more about this too, messenger RNA then actually has every 3 bases on it is a codon. Every 3 bases on messenger RNA is a literal code, so it's called a triplet code for an amino acid. And this is what in the ribosome or at the ribosome, so the ribosome is our RNA, ribosomal RNA protein structure. And then with the help of tRNAs will read these codons in order to then bring and place in the appropriate amino acid into the growing polypeptide in translation. Now, my template strand of DNA, that's a key word to make sure that you are clear on. The template strand is always written 3' to 5', and then the messenger RNA that is built off of that is then in 5' to 3'. So this is my template strand. The other side of DNA is my non-template strand. But what you can actually see with this non-template strand, the non-template strand, because of base pairing rules, actually is almost identical to my messenger RNA. The only thing that's different is I have U's instead of T's. So this non-template strand is often referred to as the coding strand as well. But the key word here is when I refer to the template strand of DNA, that's the one that we need to transcribe into messenger RNA. And we're always going to read the template strand 3' to 5' and build, because RNA polymerase, just like DNA polymerase, can only build 5' to 3'. And then when we read messenger RNA, we are also reading messenger RNA 5' to 3' is what we are doing. Now, what we'll be asked to do, and one of the skills you're going to have to make sure that you can do, is take a template strand and then determine what the messenger RNA transcript is, and then use a universal genetic code in order to read the codons and translate what these codons would code for in the growing polypeptide. What you would have to have in order to do that is a universal genetic code. So the universal genetic code is called the universal genetic code because it's universal throughout all life. Now, there are a few exceptions to the universal genetic code, but those exceptions are not anything that we are going to have to deal with. So all eukaryotes use the universal genetic code. Majority of prokaryotes use the universal genetic code too, where each codon codes for a specific amino acid. And that's what this universal genetic code is trying to show you. You will always have access to this. This is nothing you would ever have to memorize. Now, how it's written is it's written like a three-way multiplication table, is where the first base of my codon, remember my codon is every 3 bases on messenger RNA. So the absolute first codon of any growing polypeptide is always AUG. What this codes for is, it's a start codon, is what it's referred to, but it also codes for the amino acid methionine, MET. Every single polypeptide starts with this start code, AUG. You can have a huge long transcript, but the ribosome is actually not going to ever start translation until it finds the AUG first. So this is my most important codon. So then I also have 3 codons that actually tell the ribosome when to stop building the polypeptide. And again, I can have hundreds to thousands of messenger RNAs still continuing in my transcript, but as soon as a ribosome hits this stop code, one of these 3 stop codes is going to release the polypeptide and stop the translation process, is what's going to occur. Now, you can also see that in my universal genetic code and with codons, there's a lot of redundancy in here. So if I have my first codon is CC, does it really matter what my third codon actually is? Because CC, what any other base, is still going to code for proline. You look, serine. All it takes is the first 2. So we'll talk about actually what happens here with this redundancy. This redundancy actually leads to an effect called wobble, where a tRNA is actually just going to be reading the first 2 and the third one, whatever. So if I actually have a mutation that ends up affecting the third base of a codon, it often ends up being a silent mutation that doesn't change the polypeptide at all. So just be aware of that redundancy that ends up happening here. Now, here just in genetic engineering, when we're trying to actually see if a foreign DNA is red, we'll use something that actually has a clear phenotype expression. So here there's a tobacco plant that's expressing a firefly gene. Here's a pig that's expressing a jellyfish gene. So we can actually see the bioluminescence or the pigmentation that actually is a result of that. But this is just, again, evidence of the universal genetic code that DNA is DNA. So if I took DNA from a prokaryote and inserted it into a eukaryote, as long as it gets transcribed, and then it's going to get translated the exact same way. So let's kind of talk about transcription first. So we're going to talk transcription, and then we'll talk about RNA processing after transcription in eukaryotes, and then we'll talk about the translation process too. So when we talk about transcription, a term that you guys need to make sure that you are really familiar with is RNA polymerase. It is RNA polymerase that is the enzyme that is going to be transcribing DNA into messenger RNA. Now, RNA polymerase is a one-stop shop. We don't need a whole bunch of different enzymes like we needed for DNA replication. RNA polymerase is actually going to attach itself to a promoter region. There's always a segment of DNA that is upstream from the DNA that I want to transcribe that is called a promoter region. Now what's going to happen is this RNA polymerase is actually going to break the hydrogen bonds, and it's going to build an RNA transcript off the template strand of DNA. It's going to build 5' to 3' off of the template DNA. And so it's going to work and move downstream is my direction of transcription. And it's going to elongate. So first I have this initiation process where RNA polymerase attaches to a promoter region. Now, RNA polymerase is going to start building the RNA transcript, the messenger RNA, in the 5' to 3' direction until it reaches a termination sequence. And when it reaches a termination sequence, it's going to stop, and my messenger RNA transcript is going to now be ready for, in a prokaryote, it's going to be ready for translation, but in a eukaryote, it's going to need to get processed. So with this, once termination happens, my DNA just reattaches its hydrogen bonds and it's fine. And now my RNA can be used for making the protein. Now, a little more information on the initiation of transcription in a eukaryote. This promoter region of DNA. So here's my gene, promoter region included. I have my template strand, but I have this promoter region here, and promoter regions often have, in a eukaryote, they often have what's referred to as a tata box. This is this long sequence of just A's and T's that's there. It's actually a way to identify when you're looking at a sequenced genome and you go, oh, here's a long sequence of T's and A's, maybe that's a promoter region, is what that is. Now, what actually has to initiate transcription is there are other molecules that are called transcription factors. Now, transcription factors vary. There's all sorts of different transcription factors. Sometimes they're proteins, sometimes they're external chemicals. Transcription factors actually have to bind to the promoter region. And when transcription factors bind to the promoter region, this is actually the signal for RNA polymerase to attach to the promoter region and begin transcription. So what I have here is what is referred to as the transcription initiation complex. But just remember, this promoter region and transcription factors, kind of the beginning of the gene, has to be there for RNA polymerase to then start and initiate transcription. Here's just another blown up picture here, just showing you direction of transcription, where this is my template strand, and then my non-template strand or my coding strand is just there, just the other side. And messenger RNA is a single strand, and that single strand transcript is actually what is going to get used to make the protein. Now, in eukaryotes, so what we're going to talk about here is in eukaryotes, we have RNA processing. And this RNA processing actually has to kind of fix the messenger RNA. Now, in messenger RNA in a eukaryote, there are segments. So I have a start codon, I have a stop codon, and then within this is just the only section that's my coding section for my polypeptide. In RNA processing, what's going to happen is there's going to be the addition of a 5' cap. So you just need to know that a 5' cap at the beginning of this 5' end of messenger RNA, I'm going to end up having this modified guanine, this 5' cap. This is actually going to help messenger RNA escape the nucleus. This does not exist in a prokaryote. This is only in eukaryotes am I going to have a 5' cap. Now, also what happens is on the 3' end, I'm going to have a poly A tail. Now, this poly A tail is a long sequence of just adenines. Lots and lots of adenines, kind of serves a purpose similar to the telomere. It's kind of this useless segment that if it gets degraded, oh well, no big deal. But I'm only going to find a poly A tail again on messenger RNA in a eukaryote. Now, here it's kind of showing you my start codon and my stop codon, but actually what a messenger RNA that needs to be processed is it has segments of the gene that are referred to as introns. Now, introns are pieces of the messenger RNA that need to get removed. So these need to be cut out in order to leave the coding segment behind. The parts that stay are referred to as exons. So it's really, really important that you understand. In a eukaryote, we get the addition of a 5' cap and a poly A tail. But what also has to happen in RNA processing is introns have to get removed. So these introns have to be cut out before this messenger RNA can then go to the cytoplasm for translation to occur. Now, how those introns get cut out is actually pretty cool. So what I have here is this whole spliceosome. Now my spliceosome is going to splice out the introns, only leaving the exons. Now my spliceosome is actually an RNA and protein complex. And what I need you guys to start thinking about is that RNAs serve a whole lot of different purposes. And here's an example of these small RNAs. So these snRNPs, so SNRPs is what they get referred to as, small RNAs, they're actually going to be using their single strand to actually identify where is the beginning and the end of the intron. And then these other enzymes are going to actually be cutting the messenger RNA, splicing it back together, so then I get one complete RNA, where I only have exons that are there, my introns get cut out. Now, why this actually happens? Exons are actually referred to as domains. These are actually interchangeable pieces. And so what actually occurs, I can look here, here's my gene that actually has 4 different exons in it. Well, these 4 different exons can actually be spliced together in different ways. So with alternative splicing, I can have 3 exons, I can have exon 1, 2, and 3, I can have exon 1, 2, and 4, but not 3. I can have exon 1, 2, 3, and 4. So alternative splicing actually gives me variation within a gene. I kind of associate it with a chocolate chip cookie recipe that includes walnuts. So you can have a chocolate chip cookie recipe, and sometimes you don't feel like walnuts, so you take it out. Or sometimes you don't want to use chocolate chips, so you know what, I want to use butterscotch chips instead. And so it gives variation to the base protein is what can happen. Now, what we'll end up talking about in an illustrative example is we'll talk about the alternative gene splicing, the importance in our immune system. Antibodies actually come from, a lot of times, just one single gene, but now with exons interchangeable, I can actually create multiple antibodies from that one particular gene. So at this point, what we need to talk about is we need to talk about the translation process. And in the translation process, what you're seeing here is actually the process of translation where I've got my messenger RNA with its codons that are the instructions for making the polypeptide. Here's my growing polypeptide at my ribosome. So this brown blob that actually has a lower subunit and an upper subunit, this brown blob is really another RNA-based protein enzyme that is going to be initiating the building of this polypeptide. But I can't have just messenger RNA and a ribosome. I have to have tRNAs. These are really the unsung hero of translation, of building the growing polypeptide. So my tRNAs are transfer RNAs. So that's what the T stands for. So mRNA, messenger RNA. rRNA is ribosomal RNA. It's actually in here. And my tRNAs are my transfer RNAs. And what they're transferring is they're transferring amino acids to the growing polypeptide at the ribosome. Here's just 3 different models of tRNA that you guys can see. You can see it's a single strand, but it folds against each other. But the most important part of my tRNA is you see that it has an anticodon. So an anticodon is the opposite basis of the codon. So my anticodon, and because RNAs and DNAs, anytime we have a complementary strand, it has to be antiparallel. My codon, I always read 5' to 3'. The anticodon then is going to be 3' to 5'. So my anticodon is what is used to actually read the messenger RNA codon, so tRNAs know which amino acid to bring to the growing polypeptide. So the model that our textbook uses is they use this, it kind of looks like an upside down Christmas stocking, and then the fringe here is the anticodon. On the opposite end, on this 3' end of the messenger RNA, is actually the attachment site for the specific amino acid that this anticodon carries. Now, how the anticodon picks up or gets the correct amino acid attached to it is there is an enzyme here that is an aminoacyl tRNA synthetase. So this is more specific here, but this is the enzyme that is actually going to bind the amino acid to the correct tRNA. So now tRNAs that are loaded with the correct amino acid, well, they're just waiting for their codon that matches their anticodon to be highlighted in the ribosome. So at the ribosome, what I need you guys to see at the ribosome is large ribozyme, this enzyme that is actually going to help with grow the polypeptide, actually has 3 different sites in it that I do need you to be aware of. The first site to be aware of is the A site. A is where tRNA is going to initially attach to the ribosome complex. So when the codon that matches the anticodon of the tRNA is in the A site, that is when the tRNA is going to then briefly attach to the A site. What is then going to happen is I'm going to have these 2 tRNAs at my ribosome at the same time. So I have one tRNA that's already been there. It's holding on to the growing polypeptide. It is currently at the P site. The P site is where there is a growing polypeptide that is formed. Well, what happens when I have a tRNA at the P site with the growing polypeptide? I have a new tRNA that has its amino acid with it. The ribosome is going to then form this hydrolysis reaction is going to happen where this growing polypeptide is going to form a peptide bond with this amino acid. And what's going to happen then is this tRNA is then going to shift over to the P site and hold the growing polypeptide. And then this tRNA that actually just passed over the growing polypeptide to it, it shifts over to the E site. The E site is where the tRNA that's now empty can now exit the ribosome. So I have my A site where tRNAs are going to initially attach. Then the tRNA is then going to move to the P site where it holds on to the growing polypeptide until it can pass that growing polypeptide over to the next sucker who comes in, and then it's going to leave through the E site is what we should be aware of. And what moves through the small subunit and the large upper subunit is my messenger RNA is going to be moving through 5' to 3', and so my codons are going to be highlighted through each of these, well, through the A site and the P site. So here you see it, my schematic model. Here's my messenger RNA with its codons at each of the sites is what's going to be happening in order to initiate this process. Now, I can have a huge, long messenger RNA, but remember that start codon actually starts it all. And so the lower subunit is actually going to bind to this tRNA that's an initiator, this tRNA that has my MET start code, AUG, going to attach here. And this is what signals then that upper subunit to now attach. And so I'm going to have MET that starts it, and now I'm going to have my codon exposed here where my next tRNA can actually come in. And I'm just going to zip through like a little machine. You can actually see that it becomes this process of just zipping through, and I'm going to have tRNAs that are coming through, attaching to the A site, then holding on to the growing polypeptide, and then exiting through the E site is what's going to happen. This is going to occur until we hit a stop codon. At a stop codon, when that stop codon is at the A site, there's actually a release factor. And this release factor binds here, and the release factor actually kind of just clogs it up and says, release, we're done. Release the polypeptide, release the upper and the lower subunits. We are done with translation, is what ends up happening. Now, for each messenger RNA, what we typically see is we're going to see polyribosomes. And so what that means is multiple ribosomes are actually using the same messenger RNA transcript. So just be aware of that. It's not, we don't go through that whole process of RNA processing or the whole transcription process in order to just have one ribosome read it and make one polypeptide. We're going to have lots and lots and lots of ribosomes read that RNA in order to then perform translation. Now, in a eukaryote, what's going to be happening, since there's no RNA processing, is transcription and translation are going to be happening just one right after the other. So when RNA polymerases are making the RNA, ribosomes are actually attaching right there, because ribosomes, they're in the cytoplasm of a eukaryote, but there is no nucleus in a prokaryote. So we actually see the translation process happening right after the transcription process, and even before the transcription process is even completed, is what we're going to see happening in a prokaryote. So the overview, remember that transcription is reading the template strand of DNA and RNA polymerase turning it into an RNA transcript. This RNA transcript is built 5' to 3'. In a eukaryote cell, then, we have to have RNA processing. In an RNA processing, I'm going to have a 5' cap and a poly A tail added to it, and then my introns are going to be cut out, leaving just the exons. Now, my messenger RNA can leave the nucleus and can actually now, in the cytoplasm, it can actually now perform translation, where now I have all three of my RNAs working together. I'm going to have my messenger RNA that has its codons on it bind with my ribosome, my rRNA, and my tRNAs then are all going to work to synthesize this growing polypeptide. Now, last section that we need to address is we need to address mutations. Now, in mutations, because mutations happen, when I have a mutation in a gene, we need to recognize the majority of mutations that happen in a gene are referred to as a point mutation. And a point mutation means that it's just affecting usually just one base and one base pair. Now, an example would be in our hemoglobin. In hemoglobin, we talked about sickle cell anemia and sickle cell hemoglobin, where it's actually just one point mutation here that this change in DNA now results in a change in the messenger RNA codon that results in a change in the amino acid. So there's just one amino acid that's different from somebody who has normal hemoglobin to somebody who has sickle cell hemoglobin. And it's actually in just one gene. Because remember that hemoglobin actually is a quaternary structure where there's two different genes that end up coming together to form two polypeptides to now lead to this quaternary structure. So it's actually just in the beta globulin where I have this one point mutation that resulted in this one change in amino acid. Now, with these different point mutations, I have different results and different terms that we need to make sure that we're understanding. So I'm going to show you the wild type. So I'll show you what the original DNA was supposed to look like, the original polypeptide. But in this, most mutations end up being silent mutations. So in this type of point mutation where it's silent, where you're seeing the mutation is actually occurring in the third letter of a codon. And so what happens in the third letter of a codon is that wobble effect that results in no change in the amino acid because of that redundancy in our universal genetic code. Now, like we just showed you in the sickle cell, that's actually a missense is what that's referred to. It's still a point mutation, but this type of substitution ends up resulting in a change in the amino acid. So this is actually a point mutation nuclear pair substitution that results in what we call a missense. So the previous one was silent, where there was no change in the amino acids. Missense is now a change in one of the amino acids. Now I can also get a nonsense. So in a nonsense, this base pair substitution actually results in now a stop code being inserted in here. So now instead of this whole growing polypeptide, I get something that is way shorter. Or a stop code is no longer a stop code. That could be nonsense too. Now, the most, as far as a point mutation, where now is a change in one base, where it causes the greatest effect. So a mutation that is referred to as a deletion or an insertion actually results in a frame shift mutation. And what a frame shift does, if I add one extra nucleotide in here, well, now every single codon has been altered because now I added one. Well, your RNA doesn't, it reads what the information is given. So if it now has an extra base in there, every single codon from that insertion is now affected, and we can either get a nonsense or we can get something that is a complete and total missense in this frame shift, where now every polypeptide that is every amino acid added afterwards just affects the entire polypeptide, and I get something just crazy different.